Hi, how are you?
Whoops. Hello, Katerina. Hello. Hello, Serena.
Hello.
Hello, Sasirahim. I've invited you up.
Hello,
Hi.
Susan.
Hey, everyone.
Hi, Katarina.
Hello.
So I'm trying to find another, to add topics. So there's no genetics or, I don't know,
I
origins of
sent them a
life.
message. Yeah, I sent them a message about expanding the topics in science and health, and hopefully they fix it. But I sent them an email. I was like, we host a lot of rooms, and a lot of scientists and healthcare professionals can't really, have their rooms with. Yeah, so hopefully they change it.
Yeah, thank you for that. Ocean, maybe? No, no ocean
It's
evolution.
super limited. I've messaged the team as well. I haven't heard back any reply. Sasirahim, have they replied to you at least?
Nope.
Hi,
Okay, well,
Pedro.
it's time to message again.
Hi,
Hello,
Pedro.
Hi,
Pedro.
How
Pedro.
are you?
Welcome. Your mic's on the lower right, but you probably know.
No, I did not do right. I'm really bad at this. But hello, everyone. Thank you for having me.
How did the conference go?
It went pretty well. It was really nice to see some old friends after so long without going to a conference and meeting new people. Also, it was really nice. The city is also amazing, Catarina. I have
Yeah.
never been here, but it's so cute, so small, so nice.
You're in Lisboa or Oeiras? I don't know.
Oh no, I'm in Guimarães.
Ah, in Guimarães. Oh, yeah, it's very beautiful. It's the origins of Portugal. Did you go to the first castle?
Yeah, we went to the castle. We also went to that church up top of the hill where you can take the like kind of a ferry, like a cable car.
Yep, wonderful. Yeah, it's very pretty. Yeah, I'm glad you're enjoying it. It's nice. Is it very hot or is it okay?
No, it's fine. The weather is pretty good actually. It's way better than in Austin.
Yeah, well, if you live in Texas, like nothing else is probably hot anymore.
Yeah, exactly. The nights are really nice. It's like 20, 22 degrees. It's pretty cool.
I like this travel segment of Science Society. This is really fun.
Yeah, me too. What did you eat today? That's the most important part in Portugal. What did you eat? What are you going to eat?
Please tell us.
I'm 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 eating a lot of meat because I miss this since I'm from Brazil, as some of you might know. I lived in the Netherlands before, and then I moved to Texas. And Texas people used to eat meat, but it's really weird the way they prepare it. But in the Netherlands, it was pretty rare for me to eat meat. So yes, I'm eating a lot of meat around here with potatoes and rice. Yes, it's been wonderful. But I have a sweet tooth, so yeah, I'm in the right place. I really like the sweets here.
Oh yeah, they're right. The egg one with the liquid egg, the yellow one.
The pastéis de Belém.
Yeah, those two. Yeah, those are really good. They're originally from Lisbon, but they make them everywhere well now, so it's really good
Can we
here.
please hear more about that? The egg one? I don't know what that is.
Well, it's kind of a filou thing around, like a cup, and then inside is a yellow egg-based vanilla kind of pudding, and then it's burnt on the top. So how you have to do it, you have to first bake that cup, and then you put the filling in, and then you really put some sugar on it and really burn the top. So it's kind of a creme brulee in a filou cup kind of thing, to give you an idea.
Oh, that sounds beautiful with lots of really good texture.
Yeah, the good ones are really crunchy outside, and inside you have like really creamy creme brulee type of thing, like really creamy. It's so good.
Wow, we have 4 more minutes that we can talk about sweets of Portugal.
Yeah, so a lot of them are very dense. So you have, I don't know if you saw them maybe in pastries, Pedro, the ones that have yellow filling, like kind of liquidy yellow filling inside. They are it's from convents back in time, and like people back in time had mostly a lot of chicken, not too much else, like in the villages. So they made like a lot of sweets that have just a lot of egg yolk. So it's basically egg yolk, sweet egg yolk with like different things around. It doesn't sound as yummy as it is. And then you have
That
fused ovos. It's like you make kind of sweet spaghetti just with egg yolk and put it on top. I don't know if you ever tasted it, Pedro, but it's so good.
Yeah, no, no, yeah, we have this in Brazil also, fuji ovos. But the first thing that you're describing is what they call ovos moles
Yes,
aqui, or no?
ovos moles. That's the one I was explaining. It's so good. Yeah.
Yes, we have those here during the coffee breaks in the conference. They were really good.
That's a good thing about conferences in Portugal. You always have like every 2 or 3 hours you have a coffee break with something to eat, right? I don't know if it's like that, but in our conferences from our PhD program, GABA, it's always like that. Like in the morning we have some breakfast, and we have in the middle of the morning coffee and some sweets, and then lunch, and then in the middle of the afternoon again, and then dinner. So we always eat.
That's really good for your attitude.
Yeah, it's awesome. You're having little snacks all the time. It's just really good.
Yeah, that's what the doctors used to say in Portugal. I don't know if it's still the same, but you should eat 5 times a day and not too much, and not keep the stomach empty. That would be bad for your stomach. That was what I was taught all the time. I'm not sure if that's still what they teach you, but that's the idea, I guess. And don't go into the water for hours if you ate something heavy. So I would struggle with that and get out in hiding and go into the ocean, although I didn't wait the 2 hours after eating lunch.
Yeah, has been fun around here. I'm gonna. I think I told you I'm going south a little bit. I'm gonna. Some friends from the Netherlands are meeting me there. We're gonna have. We rent a house. I'm gonna. mm, Yes.
Yeah, so where are you going in the south? That sounds amazing.
Go.
Like Tejo or Algarve? Or
I'm staying in kind of the middle of nowhere, but it's 10 minutes from Nazaré. We're going to have some swim, try to surf a little bit.
okay, so you're staying in the north? Yeah, it's nice there. Just it's a little bit cold. You should bring us. That's the funniest principle that no one understands unless you go in North Portugal to the beach. If you're from North Portugal, you bring a sweater to the beach. And if you're from South Portugal, people think you're crazy, right? Unless until they go to the beach with you, and then they all regret not having a sweater at the beach. because it gets really windy.
Okay, that's good to know. But yeah, let's see.
Yeah, or you can
I have some winter clothes here with me.
yeah, you can rent a paraviento or buy one. It's not expensive. So if the wind is stopped, you're fine. It's hot. It's just by the water. There's wind, and then if you go surfing, you should really get a surf suit.
The wetsuit. Mm-hmm.
Yeah, you need a wetsuit in Portugal. Yeah.
All right.
And put sunscreen on. You will never feel you have a sunburn because it's windy. You don't feel it. You're cold and you get the sunburn.
I see that can be a problem. Can you
And
all hear me well?
yes,
My phone is a little. Okay, great.
sorry for going on and on about Portugal and the beach.
This is the
No,
best
it's great.
And we have stingrays, so
conversation ever.
be careful with stingrays. And the thing is, though, if you get stung and you feel like something horrible, it's horrible pain. But if you go to the lifeguards, there's a nurse somewhere on the beach that will give you an injection and the pain is gone immediately, like into the
Oh,
toe or whatever. So yeah, if something hurts all of a sudden horribly, it's probably a stingray and they fix it really fast right there. So
okay. That's also good to know. Thank you.
okay.
Keep that in mind.
Sorry. My brother, I'm sorry, it's the last story I'm telling. My brother, he would surf a lot. And he would have all the suit on he would get stung by the stingray all the time, and then even his feet were covered later on, so he would get stung, and then the only thing that was left was his fingers that were open, so he got rolled over by a wave and the stingray stung in his thumb. I'm sorry.
Oh God, poor guy.
Yeah, he was used to it. It just, got the nurse a thing, and that was that. Okay, now, sorry. Let's start and welcome everyone to the Science Society, and a special welcome to Pedro, Dr. Liam, who will talk about this really interesting research here today. And I'm really excited, and I know a lot of people here in the room are really excited about this research. So let me give a short introduction. Pedro Leão, he's a postdoctoral fellow at the Department of Marine Science, and he in Texas, and he did his PhD in microbiology in 2018 at the Universidade Federal de Rio de Janeiro. Sorry for my horrible Portuguese accent. And he then worked as a visiting professor at the Department of Microbiology at the same institute. And then as a FSE fellow at the University of Groningen as part of the Groningen Biomolecular Sciences and Biotechnology Institute. So his interests are around metabolic and phylogenetic diversity of prokaryotes and the evolution. He is currently, as I said, a postdoc in Dr. Brett Baker's lab at the University of Texas, where he studies the diversity of Asgard archaea and its role in the origin of eukaryotic life. So, yeah, thank you so much for coming and sharing your interesting work with us. And Victoria would ask you a couple of interview questions before we go into your talk, if that's okay. Thank you.
Yeah, sounds great. Thank you for the lovely introduction, Katarina.
You there, Victoria? Oh, she might be.
Yes. I was just navigating to my screen. Yes. as navigating back to the I'm pulling over is what I'm doing because I was driving, and so I am pulling over. Sorry. Okay, I'm pulled over. Let me turn off my GPS because it's making sounds. All right. Hello, everyone. All right. So, Pedro, as Katarina said, Science Society welcomes you. We're so happy you're here. I'm particularly excited to hear about Archaea and whatever you have to share about that. Can't wait. But to bring us into your talk, we would really love to hear about your personal connection to science, and that in particular, when you first recognized that science was something that you were feeling connected to. So if you think back, maybe reflect in your life, it could be when you were a child or any time in your life, really, an experience or a teacher or parents. But if you can tell us maybe when you noticed that you felt like a science kind of a person. And so thank you. That's my question.
Thank you. Yeah, this is tricky, right? Because we not only for science but for other professions like medicine or law or something like this, there are that type of person to say, "Oh, I was born like this. I'm always passionate about this topic. Always when people ask me when I was a kid what I would like to do, and I would say I want to be a engineer or something." With me, it was not like that because at first, in the beginning when I was younger, I never knew, I never knew any scientists in my family. There's no one that went to academia. My mom and my dad went to college even after me, or during the same time as I did. So when I was a kid, it was something really distant. But looking back now, I can remember some of my behavior that might be considered like scientific driven. Let's put it this way. For example, when I was 7, I used to swim a lot. In a swimming school, teaching how to swim. And then when I was 14, it was like, okay, I wanna do this better. I wanna understand how to make this, how to swim faster, how to be more efficient. So I started looking to videos of people swimming, the Olympics or the nationals. I went to some swim competitions and I was observing and taking notes and all this stuff. I was kind of creeping around all the swimmers that I knew. So this, I think, now looking back, is kind of a behavior of trying to observe how something is done and then try to replicate. It's pretty much what we do in science, right? We check what other groups are doing on their papers, and then we have some ideas of not only new hypotheses, but through their techniques, how to answer some of our questions also. And when I was older, I think I have a really good connection with one of my science teachers in school. And then was when I started to figuring out that, this exists a possibility of having a career doing this type of thing. So this is pretty cool. Then I remember that I was always fascinated by the illustrations in the science book, especially the ones about the cells and the interior of the cells. And this was already in high school or going to high school. And yeah, this professor opened this door to me and say, "Oh, I know some people in the university here. Do you want to visit one of their labs?" So I went there. I saw how a lab worked more precisely, and I said, "Oh, you know what? I think I would like to try to do this." And I just decided that I would like to work with science. And as I was saying, I like the illustrations in the biology books. So I said, okay, let's do something related with biology. But I always was fascinated by tiny things, like as I was saying, the illustration of the cells. I was never really understand how you could take a picture of a membrane or some stuff so tiny. So I found out this major in Brazil that is specific to microbiology. And I signed up for this, and yeah, the rest is history. I did my undergrad, my master, and my PhD in the same place, in the same lab. And yeah, now I'm here.
Thank you very much. We're glad you're here. And yeah, it's interesting to hear that you were just always observing and always wondering why. And so it's as if you've made that that your life's work, is wondering why, and taking the path that will help you figure out your questions. So can you, from that point that you left us, bring us maybe through events up to where you are now, that you've come to do this research that you're about to present?
Yeah, it's not a straight line, which makes, I don't know, makes me kind of proud of, I guess, because coming from Brazil, it's kind of hard to build up to do science in a to a broader audience. Let's put it this way, maybe. Our funding for science in Brazil, it's pretty bad at the moment. It was never really good, but at the moment they are really bad. And every time I present something somewhere, it's really nice to show people, some other Brazilians that are outside or that they can make it. It's not easy, but they can do it. We have great scientists there. If we had more funding, I'm pretty sure we would be doing way more great things that we are doing. But as I was saying, I stay. All my formation was in the same place in Brazil, in the university, the Federal University of Rio. I have one great mentor. When I was an undergrad, I already asked him to be in his lab. I remember that I looked for him 3 times because he was working the microscopy lab. And since I was fascinated by the drawings and all the stuff, I said, okay, I want to take pictures of these cells. And I was looking for him 3 times, and he was never in the lab. And on the 6th or the 5th, I don't know which time exactly, but almost in the 10th, I knocked on his door and he was there. And I remember that he looked at me and said, "Oh, you finally found me. I now have to talk to you." And I was like really scared, right? Because I was talking to a professor, never had this connection. And yeah, he listened why I was looking for him, why I would like to work there. I was an undergrad. It was an unpaid internship. And that's when it started. And I was, since this day, I stayed there till the half of my PhD, developing some research, being part of and growing the lab, right? Getting experience and getting more responsibilities through the years. And I was developing work with magnetostatic bacteria, so bacteria that produce a tiny magnet inside them and can oriented swim through the geomagnetic field, so the field on Earth, and taking pictures, doing crystallography, a lot of stuff, nothing related with what I do today. And a lot of stuff happened. There was a kind of a mess. I went to the U.S. to the University of Berkeley in my last year with a fellowship from the Brazilian government. And when I was there, my supervisor that was with me through my whole career passed away. And this was really a turning point for me because he was a really good friend also. We live in the same street. And yeah, when I was there, I was in doubt if I could keep doing the same stuff without his support, without his mentorship. But I found some amazing people that had my back. My supervisor at the University at Berkeley, Arash Komeli, he's a great guy. He's the one who put me on under his wing and say, you know what, I know you. If you want to keep doing this, I'm gonna have your back. I'm gonna do whatever is needed to you to don't give up on this. And I went back to Brazil, and that's why I had the chance to be a visiting scholar in Brazil, because I was the senior student in his lab when he passed away. And the university needs someone to first teach his classes, and second to finish orienting the students that was there, so the master students and 2 of the PhD students. And of course, the university did not have money to hire a full professor, so they did not offer me, but they opened this position. And since I was already teaching some of his classes and everything, I was really prepared to do the exam and to be hired. And that's how I became a visiting scholar. And with this position there, gave me a lot of responsibilities that it was not so used to for someone at my age in that stage of my career. But this gave me some other responsibilities that were not so cool. I had to finish my PhD 1 year earlier, so I rushed my thesis a lot. But yeah, all this experience allowed me to apply to better things later. That's how I applied to go to the Netherlands for this FSC fellowship, which is a position that it's 70% about teaching and 30% about research. And since I have a lot of experience teaching from Brazil, I was hired. And then in the Netherlands is where it started my story with Archaea. There was a professor there that I really would like to work with. Unfortunately, it never worked. She didn't have the money to hire me, and she was just starting her group. Her name is Tessa Quax, and yeah, never worked. I am still fascinated by her research. I'm a huge fan of her work, but never did not work. So I went to Groningen, was working there, and this was always on the back of my head. Oh, I want to work with Archaea. Some people at the university was working with Archaea, but I never had a chance to work with them. But I was teaching a lot, so it was just a dream or a story on the back of my head. And then I saw a possibility to apply to work with Brad Baker, my supervisor right now. I applied for a fellowship to go to his lab. We didn't get the fellowship, but he was like, "Oh, you really want to work here, right?" And I said, "Yes, I do. I really want to work with the Oscar Archaea, with metagenomics and other stuff." So that's where the stories cross. Brad worked for a long time at Berkeley, and he knew Arash, and he asked Arash for a reference. He was like, "Oh, we applied for a fellowship. He did not get the fellowship, but I feel like he really want to work here, how it's worked with him." And Arash was the responsible for giving me a really good reference and made Brad offer me a position when he got some funding to hire a new postdoc. And when I got to Brad's lab, I have no idea how to do metagenomics. I have barely no knowledge about Asgard's Archaea in general, just the general, just what I heard in the papers. But I saw it as a great opportunity. I was so happy, and people in the lab received me so well. Ian, the first author of the paper that we're going to talk today, was finishing his PhD, working with not only viruses. This story that we're going to talk today was just one chapter in his thesis. And when I saw it, I have some ideas of doing different approaches to find the same thing he was looking for. And Ian was already finishing his thesis, so just want to put this paper out. And he's not in academia anymore. He became a doctor and his desires to work in the industry. Now he's doing a postdoc again in another place, but his main goal is to go to industry. So I had a chance to approach Brad with this new strategy to find more viruses. And Brad said, "Why not? Go for it." It's part of the project where I was hired, which is a project to try to understand the origin of the eukaryotic cell. And yeah, that's when I started to develop the story that I'm going to tell you all today.
And we can't wait. This is great, and it was interesting to hear all of the connections. That even if something doesn't seem like it's going the way that you hope, then eventually it brings you here, and so at this point, you're welcome to begin your talk, and you see that your link is pinned, so you can lead people through the slides. And also, if you would prefer to have your Q&A following your talk, then that's great. We're here for you. And if you would prefer that Q&A drives the body of your discussion, then that's up to you too. Sometimes people put questions in the room chat, and we are all here to moderate that for you, so you don't need to worry about anything except relaxing and delivering your talk and letting us know what we can do. So thank you so much, Pedro.
Awesome. Thank you, Victoria, for the questions. And yeah, I think I prefer to do like a conversation. I don't see this as a talk per se. If it's not too messy, if someone had a question in the middle of what I'm saying, or if you want me to talk a little bit more about a specific thing, please let me know. I'm glad to stop and to go back to some slides that were more interested to someone. So just let me know. I'm really flexible on how this is gonna go. But yes,
Yeah, the mic is yours. Take it away.
all right. So if people want to follow through the slides, that's great. If don't, that's also great. But as I was saying in the beginning, I'm a postdoc at the Baker Lab, the University of Texas at Austin, as part of the Marine Science Institute. And the project, the funding to the project that I work on came from the Moore and Simons Foundation. So it's a private foundation that gave funding to science. And the whole idea of the project, like the big picture, is to understand the origin of eukaryotes. So how the first eukaryotic cell was formed. And one of the branches is to try to understand viruses that infect Ascaris archaea. In the next couple of slides, I will try to link why Ascaris are important for the eukaryogenesis, for the origin of the eukaryotes, and where viruses can be mixed in this story. But just to have a short introduction about viruses and other stuff. The second slide, I show microbiology in numbers. Let's put it this way. You read a lot in the microbiology papers and the stories they tell that we live in a microbial world, right? That microbes are all over. You have microbes in your skin. You have them on Earth, on water, even on the air. Nowadays, we know we have microbes. But if we put this in numbers, what this means? So we have in the whole planet Earth, we have around 7 billion people, a little bit more. We have an estimation of 100 billion fishes in the ocean, 10 to the 19 insects on Earth. So all Earth have 10 to the 19 insects. And if you go outside our planet, the estimation of the number of stars in the whole universe is 10 to the 24th. And coming back here to our microbial realm, the estimation is that we have 10 to the 30 microbial cells on our planet. And when we analyze viruses, this is at least one order of magnitude higher than the amount of microbial cells we have here. So if you see just by the numbers, instead of saying that we live in a microbial world, we can say that we live in a viral world because we have a lot of viruses around here. The picture, if you're seeing the picture in the slide on the right, I'm just putting this picture here because just so you all know, when I talk about viruses, I'm talking about all the structures. We are used to that when we talk about viruses, the first thing that came to our minds is that sphere with the little spikes, especially today with the COVID. That's the image that we see a lot on the news and on journals and everything. But all these types of structures are viruses. We don't have only that sphere with the spikes. We can have a more geometric form, have a cylinder form. We even have the phages. There's the viruses that can infect bacteria that have kind of that shape of a little spider or a little rocket or whatever, how you want to call it. So yeah, just to have a broad idea that viruses are also really diverse. We have really a lot of different types of viruses. And since we have so many viruses on Earth, going to the third slide, there's a lot of ways to study these viruses. We can study them as individuals, so just go and look to one viruses, try to understand their shape, their form, their structure, how they process in their genetic material. But also you can try to understand how these viruses are interacting with their hosts. So how they infect their hosts, when they infect their hosts, what they're doing with their metabolism. Or we can go a little further and study a population of viruses. So we have a lot of different viruses. They're interacting in the environment, how this work, what one virus is doing to the other. If I have two viruses infecting the same cell, what's going to happen? Going a little more deep, you can study the community. So how all these viruses are interacting with other organisms. They're not infecting, but they are interacting because they are sharing the same space. And in a broader view, even more broad, you can try to understand what's the impact of these viruses in the ecosystems that we have. So in the oceans, in the glaciers, in the hydrothermal vents, in the medical, inside hospitals. So yes, there's a lot of different layers that you can use to study viruses, but of course they're all interconnected. A lot of these viruses that I'm gonna talk today, all the viruses that I'm gonna talk today actually, but most of the viruses, we cannot cultivate them. So they are really unknown to us. We know they are in the environment, but it's really hard to recover them in a lab and to look through them with more details. When one of the tools that we can use to study things that we cannot cultivate, so things that we know they are present in the environment, but we don't have the tools to have them in the lab, is through metagenomics. So metagenomics seems like a really fancy concept, but if you go to slide 4, I will try to put it in a nutshell. So metagenomics is the ability to recover genomic information from samples from the environment. Basically, that's what it means. And I'll start with us going to a site, to the environment, collecting some samples. This sample can be sediment, can be water, can be soil, can be a part of a plant. We collect the sample, we go to the lab, we extract the DNA from the sample. Once you have this DNA, we send them to sequencing. So basically, we put our DNA molecule inside a machine, and this machine will read that DNA and translate to a code that a computer can read. And this code is the order of the nucleotides of that sequence. So I put my molecule there, the DNA molecule, and on the other side, I'm going to receive a file with the order of the nucleotides of that sequence. So order of letters A, P, C, G. And with this information that just came from the sequencer, we call DNA reads, these really short pieces of DNA sequences. We go through a computational approach to try to make them longer reads, so longer DNA sequences. This process we call assembly, and from the small reads that we receive from the computer, we are going to build contigs. They're nothing else more than just continuous pieces of DNA. That's how we call contigs. So we have small pieces. We put these pieces together, trying to align their ends to build this contig. So now we have larger pieces. The next step is through some specific characteristics of this context, we try to group them together. I can try to do an analogy here that I think is really good, but sometimes you don't translate that well. But imagine that we have a lot of paragraphs from different books, but they're all mixed together in the same bag. So let's say we have Harry Potter, we have, I don't know, another book. I don't know, Lord of the Rings, another book. Yes, let's say we have Harry Potter and Lord of the Rings in the same book, and we have a microbiology book, textbook from a microbiology course. And we have a lot of different paragraphs grouped together. These paragraphs will be, let's say, the context, and they are inside the same book. If I never read any of these books, but I start reading the paragraphs, I can see some patterns, right? I mean, there will be some paragraphs talking about, I don't know, wizards. And I can say, okay, wizards. So this is probably not part of a microbiology textbook, but can be a Lord of the Rings or a Harry Potter book. And then I have some other paragraphs talking about DNA. And I say, okay, there's definitely no DNA in a Harry Potter book or in a Lord of the Rings for sure. So this is another group. And then you can start grouping them together by this parameters. And then you go a little deeper, use a little bit more defined words. And then in the end, the whole thing is that probably you'll be really, really good in separating all the paragraphs from the microbiology textbook. And maybe you're going to have some mixed paragraphs between Lord of the Rings and Harry Potter. But then you can use other approaches that is not exactly the one that I'm explaining to try to separate them, and so on and so on. So we use a lot of different protocols to try to separate what we call clustering together every piece of DNA, so every one of the contexts that belong to each individual type of cell that was present in that environment. Once you can group them together, we can start to build their genomes. Because let's say in the analogy that I was doing is like each different book was a different genome. So every different book was a genome for a specific cell. So once we put them together, we have what we call MAGs, or metagenomic assembly genomes. So genomes of cells that came from environmental samples. If you see on figure 7, you will see that the circles, the yellow circle, the red circle, and the blue circle, they have some gaps, right? Because of course, if I'm trying to build a whole book based on specific paragraphs, I will be missing some paragraphs of this book. And that's okay. That's the best we can do. The parameters that we use to have a high quality mag, that's what our lab is really famous for, is that we will consider a high quality mag, and we will use them to do our analysis. If they have less than 10% of contamination, they have like 10% of paragraphs that do not belong there, and if they are more than 50% complete. So if I have a book that is more than 50% complete, and I have less than 10% of weird paragraphs that I'm not sure belongs there, I'm going to use this. This is a nice book to recover information about that subject that I want. And yes, with the mags, it's the same. Once we have these mags, we can analyze them to say, who are they, where they came from, what they can do, what this genome is capable of doing, and all this stuff. What I'm going to talk today here is viruses that infect Asger archaea. And this was recently published by us in the Breakout Lab, led by Dr. Ian Rambo, which was the PhD student at the lab at the time, and other two groups, one from the Netherlands and another one from France, from Paris. These other two works can be seen in this fifth slide. And one of them, the one from the group in the Netherlands, was led by Danny Tamarik, a good friend. And the one from the group in Paris was led by Sofia Medvedeva, which I had the pleasure to meet during this conference that I'm attending here in Portugal. But okay, so I'm talking a lot about how if you can understand at least briefly how metagenomics work, now you know how we get our samples. And all the information that I'm going to present to you here today came from this mags that we construct, right? But you might be asking yourself, like, okay, you're talking about Asger archaea. I have no idea what this is. All right, that's the moment on slide 6. If you're following these slides, if you're not following, that's all good. Don't worry. But as I was talking with you all at the beginning, the main thing on our project is to understand the origin of the eukaryotic cell, right? There's a lot of models and a lot of hypotheses on how the event that lead to the first eukaryote happened. This event we call eukaryogenesis event. The leading theory today, if you're following the slides, it's on the left on the sixth slide, and you can see the different type of models to explain the eukaryogenesis event. All of them have in common is that they take into account that an archaeon and a bacterium interact with each other to form the first eukaryote. That's what they have in common, all of them. For a long time, it was thought that the archaeon that gave rise to the eukaryote, so that participate in the eukaryogenesis event, belonged to a group called TAC. And the bacteria that was present in this event belonged to a class called alpha proteobacteria. We thought about this until 2013. We were thinking about this, and then in 2015, a group of researchers lead in the lab of Dr. Thijs Ettelman, at that time in Sweden, but then he moved to the Netherlands. Anyway, they found out this group of archaea that were named Asger archaea. We can go back here if you want to know more of why they have this name. They're not related with any Marvel copyrights thing, but they found out Asgard Archaea, and the first one was characterized with the name Loki Archaeota, and we found out that this Loki was the closest related to eukaryotic cells that was not an eukaryote. So we have a prokaryotic cell, an archaea cell, that was the closest related to eukaryotes. So we were like, okay, this is the closest one that we can recover today that is similar to the one that participated in the eukaryogenesis event. So that's how Asgars became really famous, because from the information that we have so far, Asgard archaea are the closest related to the ancestor of the first eukaryotic cell. And through more studies, now today we discover way more groups of Asgard archaea. We don't have only Loki. We have Thor archaeota, Odin archaeota, Heimdall archaeota, and we also have Skadi archaea. That now today it's the group of Asgards that are more closely related to eukaryotes. So when we talk about the eukaryogenesis event, the archaeon that participate in this event, it's really close related to what we can recover today as the Skadi archaea. If we go back to that, if we go back to try to understand what is an eukaryotic cell, the eukaryotic cell is the cells just like the ones that we have in our body, right? They're really complex cells, and if we see them and try to analyze, they have a really chimeric nature. They have some characteristics from bacteria cells, like their membrane is composed of phospholipids. The process of translation, has some similarities to bacteria. So how you start processing your DNA to go to RNA, to a messenger, and then to produce your proteins. They have some characteristics similar to archaea also, especially the replication process. But what jump our eye is what they have that are unique from them. So only eukaryotic characteristics. And this is the presence of a nucleus, and also the cytoplasmatic organization. So when you ask everybody, or what you learn in school, is that how you differentiate an eukaryotic cell from the other ones is because they have a nucleus and also organelles inside them. So basically, cytoplasmatic organization through the organelles, and a nuclei in general, because they can put in a single compartment all their genetic information. So if you go back to slide 6 and look to the models of eukaryogenesis again, you'll see that the archaeon is the blue one, and the bacteria is all the other stuff from different colors. And from the second model down, for the ones that are not following the slides, I can try to explain. All these theories take into considerations that the characteristics of the cytoplasm, so the characteristics of the interior of the cell in general, of the eukaryotic cell, came from characteristics in the archaea that participate in the eukaryogenesis event. And then the organelles were inherited by the bacteria that was interacting in this event. If the information from the cytoplasm came from an archaeon, most likely all this complexity in membranes could come from the archaeon. But maybe this is a stretch. It's just a theory, right? It's a hypothesis, and all these models that you can see on slide 6, they're all models. They're all hypotheses. It's really hard to test them. But one thing that is kind of neglected in all this scenario of the models that we have today, it's from where the nuclei came from. So where this came from. This came from the archaea. This came from the bacteria. How exactly all this thing about compartmentalize their genetic material came from. And then one of the theories that we have so far is on slide 8. And the question that would, after knowing now why Asgars are so important and their relationship with eukaryotes, you can ask yourself, what is so interesting about viruses that can infect Asgars? So in 2020, a researcher called Philip Bell came with a theory on a hypothesis, to be more precise, called viral eukaryogenesis hypothesis. So what he observed is that only some type of viruses and eukaryotic cells were capable to put in a compartment their genetic material and uncouple transcription from translation. So what he's saying here is that only some eukaryotes and some viruses could produce RNA and translate this RNA to a protein in different areas of the cell. And this area of the cell would be the nucleus, right? It would be you produce your RNA molecule inside the nucleus, and then you transport them outside, and then you translate to become a protein, which is the things that you're going to actually use in your cell. There is a type of viruses that we call NCLDVs, is nucleocytoplasmatic large DNA viruses. We're going to call them giant viruses here to be easier, because this large DNA viruses means that they are huge. So this group of viruses, they produce what people call viral factory, or VF, if you're following these slides. This viral factory is exactly what I was describing before. It's one way that the viruses are capable of uncoupled transcription from translation, one characteristic that we only see in eukaryotes. And one of these processes that they do to uncouple this stuff is to what we call mRNA capping. It's a way to modify your RNA so you can do this transportation from one compartment, from the nucleus to the cytoplasm. And this is a characteristic only present on these viruses and, again, on eukaryotes. And from this observation, Philip Bell, Dr. Bell, he came with a theory that if this thing of compartmentalize the genetic material and means to transport this material from one compartment to another, to decouple processes, are only present in giant viruses, in the eukaryote, this might have come from a giant viruses through a characteristic that we can see in eukaryotes today. And if we think about it, and in all the eukaryogenesis models that we have a bacterium interacting with an archaeum. So this giant virus has to have infected one of those. We don't have in nature so far described any giant viruses infecting bacteria. We don't have properly a giant virus infecting an archaeum, but through the characteristics of the archaea, that could be a possibility that this virus was infecting an archaea. And that's exactly what he says. Giant viruses that infect the Asgar and the archaea ancestor of eukaryotes was the responsible to bring this nucleus characteristic to the eukaryotes. So basically, the theory is that a giant viruses infect the Asgar archaea that participate in the eukaryogenesis process, and these characteristics of a nuclei came from this infection. So again, this is a hypothesis, but if this is true, the question that we have was, can we still find these giant viruses infecting Asgars now, like today? Can we still see in the environment these viruses and this type of infection happening today? And that's what made Ian look for these viruses in some samples that we have, some samples that we know they're rich in Asgars. When I say rich, I mean they have more than other places, but still in a really, really, really low amount of, if you compare the whole population from the samples, we just have a little bit of Asgars comparing to the rest of the microbes that we can recover from there. But yeah, there's a lot of computational work. Ian's data set had more than 5 terabytes, and he was able to recover more than 6,000 viruses from this environment. So all type of viruses, all shapes and colors that I show on the first slide. But from all the 6,000 viruses, he was only capable to link 6 of these viruses to Asgar archaea genomes. How Ian did this, I can't go through details if any of you want. How you can link viruses that you found in the environment to one of the mags that we have, to one of the genomes that we recover. But I'm not going to go into details at the moment. But of course, I can come back if anyone want to know how we can do this. But so Ian found 6 viruses that are related to Asgar archaea genomes in our samples, and those was the first 6 Asgar viruses described. And if you go through, I can tell a little nice story. On slide 9, if you're following the slides, the 6 viruses infect 2 different groups of Asgar archaea, Loki archaeota and Hel archaeota, right? And all this group of Asgar archaeas, they receive the name of Norse gods, right? That was living in Asgard. And Ian had the idea to give some love to the animals in the mythological Norse, in the Norse mythology, and he named the viruses through the name of some of these animals. So the 2 viruses that infect Loki, they were named. One was named Finn here, and the other was named Skull. Those are 2 wolves in the mythology. Skull, I know that it's the wolf that's gonna eat the sun before or during Ragnarok. Fenrir, I want to say that it's the son of Loki, but I'm not sure. Maybe I'm wrong about this. And there are the 2 viruses that can infect Hel archaeota. One was named Hatatosk, which is a little red squirrel. I think it's a squirrel. It's a little squirrel that walked through the tree of life and send messages through the tree to the different realms. And the other one was called Nidhogg, which is the dragon that lives below the tree of life. So yeah, it's a pretty cool story. Asgar archaea have a lot of this connections to the mythology, which is pretty cool. Makes you learn a new thing that is not related with your science at all, but it's fun. But anyway, so we have the 6 viruses, how they're named. You've just learned the story that is not in the paper, so it's pretty nice. But when we try to start studying these viruses, one of the first things we want to know is how is the protein content they have. And if you go to slide 10, I can show you some really colorful images that what it means. So the first information that we want from these viruses is to know if the proteins they have are more similar to the viruses that we can find infecting bacteria, if they're more similar to other viruses that infect archaea, or if some of these proteins are similar to proteins of viruses that can infect eukaryotes. And what we find out is that most of these proteins are very similar to proteins that can infect proteins found in viruses that can infect bacteria, which for us is not really excited, right? Because we are trying to link this infection from these viruses to the origin of eukaryotes. But if we take a closer look, we'll see that all these viruses have at least a tiny signature of some proteins from viruses that can infect eukaryotes. And this is really unique because for the first time we saw viruses with this chimeric way too, with characteristics of viruses that can infect bacteria and archaea, but also eukaryotes together. Even if it's a tiny signature, this was pretty cool to watch. But the majority of the protein content of these viruses were more related to a really well-known family of viruses that infect bacteria. That's the Caldoviralis family. But so you would say, so if they're so close to a really well-known family, they should be viruses that are really similar to the other viruses that infect bacteria. And that's that on slide 11. If you see, this is a clustering process. So basically, each one of these dots that you can see, it's one of the 6,000 viruses that we will recover from the samples. And through the protein content of these viruses, so to the type of proteins these viruses can produce, we can cluster them together by how similar this protein content is. So how similar all the proteins that this virus could produce are. And the arrows here you can see for the people without the without the slides, I will try to describe this, but we have a lot of dots, so a lot of viruses in these samples that belong exactly to that family called the Correlates. That is the family where our viruses are technically more close. The protein content of our viruses are technically more close to. But even from these viruses, this called the Correlates viruses, in the image is the red, is the green dots. Even these viruses do not cluster together with the Asger viruses we found. The Asger viruses we found actually don't cluster with anything. They are like little dots that are apart from all the other dots that represent the viruses. So this is another indication that these viruses are really unique. And this cluster did not happen like this. They have their own little groups, probably because of this tiny eukaryotic signature they have on their genomes that make them really different from the other viruses that we found, even in the same sample. If we go after knowing a little bit more of the protein content of these viruses, the next step would be to do what we call an annotation of these proteins. So to try to understand what these proteins were doing, or how once you interact with their host. So once they infect the cell, what they actually do through their cell, to their metabolism of that cell, and this is in figure 12 now, in slide 12 now. And this is a really complex figure, but if you go to the slide 13, I can try to highlight a little bit of the most exciting things that we found. For example, there is some viral proteins that once these proteins are expressed by the viruses after the infection inside their host, so inside the cell they infect, they can mess up or they can influence the transcription of this host. So one of these proteins had a domain. So one piece of these proteins had a domain called homeobox domain. And this was the first time it was characterized in a prokaryotic viruses. This usually is really common in eukaryotic viruses, but not in prokaryotic viruses. Why this is so exciting? Why we were so excited about this? Because why a protein that would have an influence in the transcription process in an eukaryote would be found in a prokaryotic viruses. So if this protein is useful for this virus, it means that some parts of the transcription process in these Asgers they are infecting are more similar to the ones in eukaryotes, because this is what virus from eukaryotes use to induce the transcription in these cells. So this was pretty cool and a pretty nice indication that these viruses have eukaryotic signatures. Another protein that is pretty interesting for us is a protein modification system. So protein modification systems are systems that once your protein is ready to be used, they do little modifications on the protein, addressing a different function or addressing a specific site that the protein should act. And this is called a ubiquitin system. And again, this is a protein modification system found in eukaryotic viruses and also inside eukaryotic cells, even when they're not infected by viruses. But it's something related to eukaryotes also. So this is another indication that these viruses, they are infecting a prokaryote, right? An Asgard archaea, but even though they have some characteristics that are useful inside a metabolism of an eukaryotic cell. And this is really exciting. Another thing that we can do that, I don't know, again, if you are interested or not, but, and you can see on the paper is in slide 15, is that some of these viruses have a polymerase B, DNA polymerase B. And this is an enzyme that is really important for the DNA replication of the viruses. And some of the viruses do not have one of this on their genome, and they just use the one from their host. So once they infect the cell, they can use the DNA polymerase from the cell. But some of these viruses, 3 of them to be more precise, they have their own polymerase. So this gives these viruses a little bit of autonomy during the process of replicating their DNA. And one pretty cool thing that we can do is that this protein is really conserved. So the way the protein is, the sequence of the protein, they are conserved through different lineages of the cells. And so through the sequence of this protein, through the amino acid sequence that compose the protein, we can try to trace back from which other proteins like this with the same function these proteins are more close related to. So when we do this analysis that we call phylogenetic analysis, we can see that the protein, the POB found in fan here, for example, it's more close related to POBs that we can find in bacteria. The one that we found in Nidhogg are closest to other POBs that we can find in viruses, and the ones in Hatatos are more close related to the other POBs that we can find in Asger cells. So the conclusion that we can draw from this is that these viruses are acquiring proteins or from all over the tree of life. They're not only stick. So not all POBs from Asger viruses coming from the same place. And this is pretty cool. And to end up, now when I jump in the story, I help Ian with some of this analysis, the previous analysis, especially with one that is not here, to be fair. But what I'm doing now, and it's not in the paper yet, is to using a different approach, the approach that I skipped, that I told you how to. So Ian was using one type of approach to link the viruses that we found in the environment to the Asger host. I'm using a different one, and I'm using a little larger data set. I'm using the same data set from Ian and another collection of samples that we have from the same site, from this hydrothermal vent in Guaymas Basin. And through this new data set, together with Ian's, we were able to recover more than 10,000 viruses this time. But using this different approach, now we have 45 Asger proviruses. And from this 45, 43 are unique because 2 of these viruses, I was able, using a different techniques, to find the same viruses Ian also found in the sample, which is amazing. And what we are always looking in science, it's a way to check if our method is working. And since we are using 2 different methods and we could find the same viruses, it's a really good indication that we know what we are doing. We're doing a nice work. And most important is that from this 43 unique viruses and 41 new viruses that we recover, 26 of these viral genomes are complete. This is really valuable for us because this tells us that all the genes that virus had are there. So you can go gene by gene, try to understand their function, and try to understand what they are possibly doing to make it possible an infection, a production of a new viral particle once they infect the cell, how they're infecting. So having complete genomes is extremely valuable in metagenomic analysis. And on slide 17, if you want to take a look, I'm showing a glimpse of this 26 complete viral genomes. We're going to release, I hope soon, in a new paper. And on the left, I have a graph. And for all of you not following through the slides, it's a bar graph where I show the percentage of the proteins inside each one of these genomes and their function. And the last bar in the graph, it's a dark bar. And I can guarantee you that in all the genomes, except from one, this bar represents more than 40% of the genome content. What I'm trying to say is that all of these viral genomes, more than 40% of their proteins have unknown function. So if we do our job and try to annotate these proteins, try to find a function through homology, to comparing to other known proteins, more than 40% of these proteins have no function, no known function at all. So we never saw this in any other type of organism or any type of viruses. We don't have a really match that we can say, okay, this is more similar to this type of protein, so they might have the similar function. So no, more than 40% of the protein content in these viruses are unknown, which is pretty exciting because we can find new things, we can propose new things or new functions, or sometimes they have the same function of a known protein, but the sequence is so different. And this is also really exciting to see, because probably if they have the same function but really different sequences, this means they came from different places. And this is what drives us in the lab, and what I'm really like to try to understand where things came from, right? Not only where the eukaryotic cell came from, but where the specific process inside one cell came from. This was inherited from another cell. This was developed by a lot of different mutations that happened. So all these origin stories and this phylogeny, where things came from, are something that are really valuable questions for us in the lab. And yes, that's the story of the paper. We are trying to find this giant viruses that infect an Asgard archaea and possibly have some influence in the origin of the nuclei in the eukaryotic cells as we see them today. And if you are in the last slide, you can see a nice slide with all the members of our lab. In this paper specifically that I was talking about, Dr. Ian Ramble was the main lead of this paper. Brad was Brad Baker is the corresponding author, and Valerie De Anda is another postdoc in the lab together with me, and Marguerite Langwing it's doing her PhD now in another lab, still work with us really closely. And she's not here in this paper, but I have to thank Katie Appler for all her work putting this max together and being super helpful to all the work I have been doing to recover these new genomes that are not published yet. But yeah, that's it. If you guys want me to explain a little bit more something, if I was too fast, please let me know. I'm open to questions or to anything.
Yeah, thank you so much for this amazing talk and work that you're sharing with us here today. It's so impressive. It's like um if isn't it comparable, maybe for other people that are not in biology, if you just look back in time and space, you're kind of doing the same thing just with evolution and about life. So would you say that's a good way to put it?
Yes, it is. It is a good way. It's like, yeah, it's trying to find pieces today of something that happened so long ago. So it's really a challenge to try to tell a story. I think it's the same thing. Maybe a good comparison is with people doing archaeology. So maybe it's like you find pieces of stuff that exists in the past but do not exist right now, and you're trying to tell a story of how that thing became that thing and how that thing was lost through the time. So yeah, I mean, I think it's a really fun work to do, but it's I understand can be a little frustrating because you always have missing links. You always have things that you cannot explain how that thing emerged, how that thing was gone. So, but yeah, it's a good analogy. Yeah, we're trying to look to the past, try to understand where we came from.
And it's so amazing, that recent technology enable you to do this work, right? I mean, it probably took, you couldn't have done this like 10 or 20 years earlier. So I'm really looking forward to what, technology, data, the ability to go through more data will discover, because it's really impressive that, you know, they have so many mutations and so much happened in this millions of years, and we can somehow still trace back some of these features that were developed. It's so impressive to me. It's unbelievable. So yeah, thank you for sharing that. And please, everyone, if you want to ask a question, please flash your microphone, and yeah, go ahead and ask away. Yeah, go ahead, Eli.
So I was wondering, were there any attempts when you were recovering these to filter for viral particles and look at the morphology, even if you can't cultivate them?
Yeah, so our during our approach, we're not filtering or trying to separate the viruses from the cells. We are just extracting DNA of everything, and through the assembly process. So after we already have our sequences from the DNA sequencing, then we were trying to find specific characteristics of viruses to build these viral genomes. But no, we did not try to cultivate them because Asgard, Archaea, they are not really nice to cultivate in the lab. So far, we only have one member of Asgard, the Loki Archaeota in culture, and it's kind of in an enrichment culture kind of thing. So when we say culture, it's like usually a pure culture. It's just that cell growing in that media. And when we say enrichment, is that we have mostly that cell, but we have other things together also. So yeah, Asgaricia are not so nice to grow in the lab. They cannot grow in the lab. So we cannot isolate the viruses, right? Because to isolate the viruses in the lab, we need their host also in culture, so it can infect them and then produce more viral particles and everything.
And so they're still unculturable.
Yes, we don't have Asgaricia or these viruses in culture. No.
So just a quick mention. In the late '90s, I was in a discussion with a few people about the unculturability the unculturability of the host of rosiophages, and in that instance, which probably isn't the main story that you're up against. My suggestion was, well, maybe try culturing them at the pressures they exist at. And a year or two later, somebody at Max Planck actually did that, and they were able to culture Rosia bacteria and Rosiophage.
Yeah, cultivating Asgard, it's a big thing. People, a lot of groups are trying to do this because you can imagine that recover information from them through sequencing of a pure culture would be way easier than to do metagenomics like we're doing. But just to give an example, this group in Japan that have this Loki Archaeota in culture, they they found out that this first culture they had the doubling time of the cell. So from one cell become two. So just for them to divide, it would take 3 years, 3 years to double time. Just to have a reference, and E. coli replicate like the doubling time is like minutes. So it's really fastidious, and the conditions you have is really specific. And even this one that is in culture is not in the best scenario to build for example for the viruses to be a reliable host to cultivate the viruses, and also to develop molecular tools so you can know a little bit more about the genomes and all this stuff. But yeah, I mean there's a lot of people trying to do this at the moment.
Yeah, that example. It really ties back to why I suggested high pressure. Not that it was like anybody taking up my suggestion at the time, because pressure can affect protein structure, and protein structure can affect kinetics. And if something is taking a year to divide, when other things in the ecosystem have much faster life cycles, that really suggests that something like pressure, something really basic to the physical chemistry behind the enzymology that would affect metabolism and replication, that just I mean, that really just jumps out at me as a possibility.
Yeah, this is a long running discussion in microbiology, right? Exactly what you were saying. Even when they can grow in the environment, we always have the question or the chip on our shoulders saying, "This is exactly what I would see in the environment." Most likely not, right? Because even if they can grow in the lab, the conditions are really, really different. Even for organisms that you can find in different places, right? You can see completely different phenotypes, even sometimes different shapes if you isolate them from an ocean sample or from the sediment, for example. But yeah, this is always a question that we have, even with the ones that we can cultivate.
And similar, you mentioned enrichments. Having the right consortia. I mean, something might just limp along, and barely survive without some other organism that it forms either lifelong intimate associations with, or periodic close associations with. And just isn't there in the attempts that have been made, would be another thing that is potentially an important part of the story.
Yeah, absolutely, absolutely. If you, I don't know if you have access to the slides, but if you want the contact, I can send you the paper. One of the socio-ecological model take into consideration exactly this, the E3 model. That before they have the endosymbiotic event for the eukaryogenesis, the cells were already really close together to the other ones, and one was using the metabolites of the other one. They were exchanging things, just like we see a lot of bacteria doing nowadays in their little communities or building multi-species biofilms. So yes, definitely one of the main reasons. My guess is that one of the main reasons why we have the enrichments today is because of this, right? They're been living together for so long that everything's in a right synergy, that one is producing something for the other one to use and the other one. So if you take them apart, they do not have all the components they need to survive by themselves.
Another thing that I'll mention is the highest photosynthetic bioproductivity measured in any lab still, I believe, is Pert et al. 19. I think it's et al. 1983. It might have been him on his own. Where it was a chlorella species, but also three other prokaryotes, that is bacteria. And the association led to like 50% higher bioproductivity than just the isolated microalgae alone.
Yeah, yeah. This is something fascinating, but yeah, it's something really, really complex. When, as I mentioned before, I was working during my my PhD, with magnetostatic bacteria, with this bacteria that produce magnets. They are also really, really hard to cultivate in the lab. And yeah, we have some cultures today of these other ones, but yeah, I have been to this side of trying to cultivate things. I have to say that I prefer to stay now in the metagenomics area. It's a lot of work that need to be done, but it's a lot of hard work.
I mean, I just wanted to know what the morphology was when I first asked the question.
So I
What
can I
what do
can
these things look like?
I can link to you somewhere the paper from this Japanese group. It's a weird morphology. They actually have some like tentacles or something like this. But again, as we were discussing in the beginning, Eli, a lot of people are skeptical if that is exactly what we would see in the environment, or if some artifact of the culture, or even some artifact from the microscopy that was done. So as you can imagine, something like this, that a lot of trying to answer a big question, you have a lot of people that are skeptical, and I completely agree with it. Have to be really careful with what you are claiming, right? So, but I can link you the paper and you can see the morphology.
I'm curious. I had a question. It's fascinating about the quantity, the variety of viruses. 10 to the 31. That's spectacular. And then your slide on 17 that revealed how incomplete and how much more variety of structure of unknown function there is. I'm curious if, and I haven't looked in detail about origin of viruses, but it would. Could you comment on? Is there I mean, I would imagine there would be, but an ongoing process of originating new viruses went through either decay of detritus in an ecosystem in capsids, taking in new material and that sort of stuff. It's a sort of an ongoing process of new viruses for there to be so many and such a rich variety. Do you have thoughts on that?
Yeah, this is a great question and really interesting. First, we have to think about something a little bit more, I don't know, more simple, like what we will classify as a new viruses. What type of differences we have to have from one virus genome to another one to say that they are different. If we have a mutation in a, let's say, in a structure protein, like a protein from the capsid, that is a really important part of the viruses. That's what we would call a different one. But if we have the same type of mutation or the same size of mutation in a protein that is not structural, but maybe in a protein that is related to a more common process, this also would characterize them as different viruses. I don't know how to answer your question because phylogeny of viruses, so divide viruses in these groups, in these families, is something really messy that I'm going to put myself inside this after participating of these conferences of viruses, but we that study viruses, we are still debating a lot on how to do these classifications. A lot of the proteins in the viruses, as I show in the figure on slide 2, a lot of the protein on the viruses, even the structural ones, they're not really conserved. As you can see, we have different structure of the viruses, so these proteins can change. But answering more precisely your question, I'm pretty sure that we have the formation of new viruses all the time. The how this happened, I this is really fascinating to me, to be honest. How a complete new viruses emerge, or if you want to go back to a more simple way to view, like how the first viruses came, because it's just like the egg and the chicken, right? It's kind of not really possible for a virus to exist without a cell, but at the same time, it's possible that a virus exists before and then encounter a cell to multiplicate. So it's a really tricky question. On my theory, my personal theory is that when all these biomolecules were getting formed and we don't not even have any type of cell or anything, we have these concentrations of biological material in a lot of different ways. And at one moment, one of them put together enough things to replicate itself. And then we start calling this one a cell and all the rest viruses. And then some of this that could not replicate themselves start to learn to merge to this one that could replicate and use the system to replicate. And then I guess the first viruses came. But how this process is happening right now in the environment, I don't know. I have no idea. But definitely is happening.
I mean, it's curious because in terms of how you might age viruses in the sense or date them as is, I mean, would they necessarily, if they're in some sense incorporating sequences from decaying either eukaryotes or bacteria into capsids, that might complicate the analysis of the taxonomy, right? I mean, in the sense that, I mean, I guess it's a question of how you might date them, uh, and whether they would be acquiring sequences that are much older than them through some other environmental mechanism.
Yes, there is two. So if in slide 11, when I showed that clustering using the whole protein content, this is usually how we try to do this let's call classification of these viruses from environments, because we're not necessarily saying where they belong to. We're just trying to closer them together to see what are the type of viruses that are more close related to them. And this is why we use the whole protein content, right? So if they acquire one protein from one bacteria and another virus acquire just one protein from eukaryote, they're gonna still cluster together because it's just one protein. So all the other proteins are together. And one important thing, I think, what you were saying about aging, please correct me if I'm wrong, but they cannot accumulate a lot of proteins that they are recovering from different hosts or from different cells, because the capsid limits the amount of DNA they can put together in one particle, and they have those genes that are really necessary for them to produce a new viral particle. So what I'm trying to say is that if at one point you accumulate too many genes from your host, you end up losing some genes that might be really necessary for you when you infect another cell, you be able to produce a new particle and infect another cell. So there are a lot of new phages, new viruses being formed, but also we have to keep in mind that there is a lot of viruses that exist in the past that do not exist nowadays, because they lose this possibility through mutation or to letting go some of the proteins that were really important for them to produce new particles and to infect new cells.
Yeah, thank you. It's interesting. It's fascinating research. I really enjoyed your talk. It's exciting to see that there's so many proteins of unknown function to discover too. Thank you.
Thank you so much for coming around.
Yeah, thank you so much, Pedro. That was really fascinating work. And first of all, I was thinking about the biohybrids because based upon whatever you describe, it's, I mean, it's we can just do it in a lab based upon the biohybrids. But another point that it came to my side that was about the ubiquitous system that you just founded. During your research, and I was just wondering what thoughts you have around that, because when we want to think about that, there is a lots of potential come around. For example, about the viral regulatory that you just explain about that, or we can think about the tumor necrosing factor and even the actin filament, and everything can be related to this system. And I was just wondering, after finding this system, what was your hypothesis goes?
Yes, thank you so much. That's a great question. As I was saying, it's really interesting for us because it's a possibility to, it's an even more strong argument to link Asgard's with the origin of eukaryotes, right? Because if we're seeing something that is present in a eukaryotic cell and a virus is infecting a prokaryotic cell is using this system, it means that the system should be something important for the metabolism of the host. And my theory specifically about the participation of the, no, not my theory, it's not my theory, but the theory that we are trying to prove here, that the viruses were participating in the karyogenesis event, it's awesome, right? Because we are linking them somehow. But it's even more important for the theory that puts the Asgard as the closest related to the Archean that participate in the eukaryogenesis event. Um, the ubiquitin system proteins, they are part of a family of proteins that we call ESPs. Those are the eukaryotic signature proteins. So these are proteins that are specific from eukaryotes. And when we are looking through Asgard genomes, not only the viruses, we are always looking for this family of proteins, because we want exactly this proteins with signatures from eukaryotes to be found in this Asgard archaea. So that's what is really important to find this in viruses also. But the implications of this through the not to the virus, I can answer your to you what I think, which type of processes they are doing. But I can tell you that the implications of the presence of these proteins in Asgard is a really strong argument that eukaryotes came from a similar Asgard archaea through the eukaryogenesis process.
Thank you so much. I see that Katerina just you open your mic. Maybe you wanna ask a
Oh
question.
no, go ahead. I'm sorry. I wasn't.
Yeah. Thank you so much. Thank you so much for explanation, Pedro. But that was really very interesting part to me, and I think that there's I mean, lots of potential out of this research. Thank you.
Thank you for the question. That was great.
Yeah, I see Frank, Nick, Dennis. Please go ahead. If you, Pedro, I want to also check with you how much time you still have with us. So do you still have time for like 3 or 4 questions?
Sure, no problem.
Thank you.
Oh, hi, Pedro. Thanks for sharing this very interesting research with us. Quick question regarding the thermal vent that you get the sample. So, what is the significance of this particular environment? Is it could it be so I guess my interest as a layperson is the more general question. So what is what your findings in this research has implications on, for example, what Serena referred to, the origin of whatever, right? Origin of viruses, origin of uh, archaea, for example. The is it can I say this is put DNA more to the first and then RNA? Is that a relevant question? So I guess I was asking two. I'm asking two questions. First is the thermal vent, and then the other is the origin of DNA or RNA.
All right. About the hydrothermal vent first. This site is a really unique site because the hydrothermal vent. If people are not familiar with hydrothermal vents, it's just like on the bottom of the sea. We have this little openings that keep floating. Sorry, that keep floating a little bit of water. They keep cycling the water. So it's a really dynamic place. And in Guaymas Basin, so this hydrothermal vent in Guaymas, it it is special because when we do metagenomics there, we can find all sort of things. And so since the environment is so dynamic, it's changing all the time the type of nutrients that are available or the types of minerals. And sometimes the hydrothermal vents are active, sometimes they're not. So this makes the community there really not only rich because also they have a lot of carbon in there, so a lot of food for the microbes, but because they have to keep their metabolism always up so they can turn up to this all this dynamic that is happening there. And also the diversity is really big. You have everything that you can imagine in these samples. And the second question, you were talking about if there is any implications in the origin of DNA or RNA through this information that we have. That was the question. I don't remember.
Yes.
Not really. Not really what we were focusing or what we were looking into. And I'm not sure how I would link these two things. But this is a really recurring question in biology in general. There are some theories that says that the RNA came first. So the first cell, the first biological entity, let's put it this way, had stored their information through RNA, and then later they start to see, like, through mutations and through some other cells, that DNA were more efficient. But I don't know. There's a lot of theories the other way around also, saying that all the systems became through store information in DNA form, and then the RNA came later. But no, in our research, we are not trying to focus on understand this.
If I may add, we had a guest speaker here a couple of 3 months ago, that he found that actually proteins can form on stardust under very regular conditions. It wouldn't be even anything special. So that's another new insight that we have nowadays. But this goes a little bit away from the topic, but I just wanted to add that. Thank you.
Yeah, that's really interesting.
Yeah, go ahead.
Hi Pedro, I was curious why you chose the Guyamas Basin and whether there have been opportunities to, one, like how many other basins are in maybe like North America that might be suitable for this sort of sampling, and if there have been anything of this nature taking place at those. I'm curious about the larger picture.
Yeah, thank you, Dennis. The Guaymas Basin is a really well characterized site for research, and this is really important for us to have a lot of long time data about the physical chemical conditions there, also how the diversity was before. So for us, since we cannot reproduce these experiments in the lab, it's really nice when we have a place that have been sampling the samples have coming from, and we have information through time if that place is changing too much, if the place keeps a little bit more stable. So this is one of the main reasons why a lot of the researchers are doing in Guaymas Basin, because people have been doing research there for a long time. But another really sincere answer to your question is because this is one of the places there's easier to get funding to do our research. So the collection of the samples are really expensive and really complex because it's really deep on the sea. And yeah, but for example, this projects we use some of the infrastructures from the Woods Hole Oceanography Institute. So I know that a lot of people are interested there. So it's easier to put together a crew of scientists to write a grant proposal to go to the same place. But I'm pretty sure there are some other studies in other basins. I know in the US, I'm not sure, but I know that in China they are. In the North Sea also they are. So to answer your question, it's a place that was well studied, which is important for us to have more information. And also a lot of people are interested in that, so it's easier to grab scientists to get funding to go to this type of places.
That all makes sense. Yeah, good baseline data really helps when you're doing longitudinal observations and to be able to map the changes. Thanks so much.
You're welcome. Thank you for the question.
Nick and Parth, please go ahead.
Yeah. Hi. Thank you for the lecture. I have a very lay question which might require a lot of time to answer, but even answer in principle will be satisfying to me. So let's say you have managed to sequence the DNA or RNA of a virus or something else. So you have a chain of bases, DNA bases. You can slice this chain in different ways. Maybe you can use 100, 200 or 300 sequence bases or less. So my question is, how do you know what slices of this DNA sequence is a gene? Obviously, how do you determine which parts of it are active genes, which are not active genes, junk DNA or RNA? Like in physics, for example, you have a theory The theory has predictions. You do an experiment, and if the experiment matches the predictions of the theory seems to be correct. How do you do this in biology, especially in the area of figuring out exactly which, the map of the genes of the genome?
Yeah, when we sequence DNAs, after we do all that process that I was trying to describe briefly through the metagenomics, to predict what is a protein, what it's RNA sequence, and all this stuff, we have specific signals in our sequences. So, for example, before we know what is a protein, we have a region called open reading frame, or ORF, that to that region produce a protein, they need to have, for example, star codon. So they need to have a one thing that we know it's a signal to start the RNA production. And if we have that sequence, we also need to have another specific region, a signature region, to once that RNA is produced from that DNA sequence, starting with the start codon. Right after the start codon, we need another specific sequence that once that is an RNA, it will be a binding site for the ribosome that we call ribosomal binding site, RBS. So we have all the signals that are necessary to things to be transcribed or translate. So the enzymes only recognize these little pieces of these DNA sequences. So those are the signatures that show to us that this is the start of a gene and this is the end. Are the same sites that the enzymes use to recognize, to start the process, and to end the process.
Okay. Okay. So
We
is it more
have
difficult?
signatures to we have signals to understand where a gene is starting and a gene is ending, basically.
Then you can determine the genes, but you would not know the function of the genes, right? Unless you do some other things.
Yes, to know the function of it. This is really, I have to be really careful with my words here, because otherwise, if some experimental biologists are listen, they're gonna kill me. So, doing metagenomics, we will never know the function of the gene. What we're gonna do is a prediction of the function. Because to know the function, scientists will tell you that you need to go to the lab, to the bench, and do experiments, right?
Oh,
But
like in
yes,
physics. All right. Okay.
yeah, but to do the prediction of the protein, like to see potentially what the protein can do, after all this, after we identify these regions that we consider a protein through the signal sequences, we do what we call annotation. So we compare the sequences to a database of known proteins. And if we have a high identity to one protein that was already tested in the lab, their function, we say, okay, we have this catalytic site, we have this binding domain, and everything that was also conserved in this protein that was already known. So we predict that they have a similar function.
Okay. Okay. Excellent. Thank you. And I assume the way you describe this thing, I assume coming up with a sequence of the DNA or RNA bases is much more difficult and labor intensive and statistical than just slicing this chain into genes without figuring out the functions of these genes, which require many more things. Is this correct or not?
It is, especially so when you do the sequencing, that is kind of like the slice process that you're describing after we extract the DNA. If you have just one organism, so if you are extracting the DNA from a pure culture where you know you only have one genome, this is way easier because it's just like building a puzzle, basically. At least to have the sequence, right? Not to predict the functions and everything. But in our case here, what I was trying to explain with the analogy of the book is that we have a lot of genomes all mixed together.
Yeah.
So to address from which one which piece of DNA came from, it's really really hard work, and a lot of computational power is required to run all this pipelines that we developed to try to cluster them together. But yeah, you're right.
Okay, thank you.
Welcome. Thank you.
Hey, Parth. Well,
Hi. Thank you for doing this room. Thank you for giving me the opportunity. Congratulations to Dr. Baker's lab. Pedro, thank you for being here. I'm fairly familiar with Dr. Baker's work. In fact, I have met his group at the Texas ASM meeting in Corpus Christi a few years ago, where I think somebody from his lab, I think he was there, if I'm not mistaken, he gave a talk on Asgard Archaea. And I've been following, we have been following your papers regularly. And thank you for the congratulations on this paper on viruses that specifically are that are specific to Asgard Archaea. So I don't have any specific question for him. I just came up to say hello and greet him, and thank you for being here. But the other reason I came up was I heard talk about RNA or DNA origins, right? So there are. I just wanted to, if the mods will permit, I'll just take 30 seconds to explain the current hypothesis, the existing hypothesis. So there are two main hypotheses. One is the hydrothermal vent origins of life, which is probably going away, as multiple lines of evidence seem to suggest that life probably emerged at the interface of land and water. There's a phenomenal paper by Jeffrey Bada from Scripps Institute that talks about islands that probably existed, small islands that probably existed around the time of the late Hadean and the primordial world, and work by Bruce Dahmer and Dave Deemer from UCSC Santa Cruz. They have shown that polymerization can happen through wet-dry cycles, along with the work of Nick Hudd and others from Georgia Tech. So that is the current emerging hypothesis, is that polymerization in the primordial world was very much could have only been possible at the interface of land and water, because you need to have this diurnal cycles of wet and dry cycles. Plus, you have an inventory of amino acids that are already being delivered to planet Earth from meteorites at the time. All these things combined, now from we have moved away from RNA world to a mutually coexisting RNA peptide world, where RNA and peptide stabilized each other and contributed each other to each other's existence, and that is how the primordial ribosome was formed, which is what we work on. So that is the gist of it. So if you want, I'll be more than happy to share papers. The last 3 years, there's a fascinating series of papers that have come in this particular area of research. Thank you very much. Once again, congratulations, Dr. Pedro.
Thank you, Purdy. That's fascinating. Also, that's something that I'm not catching up with the new theories. I have been diving down this virus rabbit hole. But yeah, I'll be happy to talk with you more about this. And it's so nice that you had the chance to listen, Brad, talking about Oscars and meet some people when we were down in Port A and Corpus. I have my email, and if you have access to the slides, I have my email in some of the slides. Also, I'm on Twitter if you want to keep chatting,
Sure,
exchange
sure.
some papers.
I'll
Sure.
probably find you. I'll probably find your email and then probably send you an email. And so I have just one comment more than a question about Asgard. I think it'll be incredibly fascinating once we have the. I think there are two genomes that I've seen. One is the Lokiarchaeota MKD1, also known as Promethearchaeum, right? That's a complete genome.
Yes.
The other is the Odinarchaeota LCB4. These two are the reasonably. The Odinarchaeota has 9 contigs. It's not complete yet, but a quick look at these two genomes would reveal some interesting features. I did some analysis of this. We will probably get a paper out in the next couple of months. I'll keep you posted. And what would be interesting is to see how much of this genome shows signatures of horizontal or lateral gene transfer from bacteria, and how much of this show are conserved of within other archaea, and how much of the remainder is shared with eukaryotes. That would be very, very interesting when we take a hard look at deeper look at the genomes. So that's the update. So we are definitely interested in this particular area of work, and I'll stay in touch. I'll find your email, and I'll definitely email you our work. Thank you very much, Dr. Pedro.
Awesome. Let's keep in touch. And just to highlight something that we have been doing in the lab. So now there are around, I want to say 100, but probably it's more. But there are some, yeah, there are 100 and something Asgard mags available. So this metagenomic assembly genomes, and part of Katie, our PhD student, part of her thesis is to characterize this new Asgard mags from Grimus, and she's presenting a part of her work in ISME in a conference for environmental microbiology next month, and she already characterized more than 500 new ones. So soon, you will and everyone will have a database of Asgard archeo or Asgard genomes that will be at least 3 times higher than the ones that we have today. And this type of analysis, the part was talking about, they're going to be possible to be done, and really excited to find out new things on these genomes, where these genes came from, to have horizontal gene transfer, all these things.
Absolutely spot on. I couldn't agree with you more.
I just wanted to mention thanks, Parth, for that rendition of contemporary thought on wet-dry cycles. I've always been an enthusiast of the role in, particularly the role in the origin of the RNA world. Always been a river shore clay wet-dry cycle enthusiast. And I'm glad to hear that it's gaining ground. I didn't mention it, Pedro, earlier because I noticed that your take sort of entered in the origin of eukaryotes, and particularly, what was the mechanism of that symbiosis, that mitochondria nucleus, and how that evolved. Um, but yeah, I maintain a deep curiosity in earlier origins of how archaea and originated from that RNA world. So this has been a fascinating discussion. Thank you.
Yeah, I also would like to add a quick point to what Serena just said. So there is a recent paper by Steve Benner's group from the Foundation for Applied Molecular Evolution based in Florida. It's like a path-breaking paper. Actually, there are 3 papers, one from his group, where they have shown that polymerization of RNA monomers is absolutely possible. I think they have done it on silicate or boron, I do not know. So what they've done is they've actually shown that polymerization is possible on crystals, number one. Number two, there are 2 papers from Ada Yonath's group in Israel. Ada Yonath is the Nobel laureate who won the Nobel Prize for solving the ribosome structure in 2009. Her group has shown that the smallest part of the ribosome, which is does the catalysis of the peptide 1 formation, it can actually be segmented further to the smallest component possible, and it will still be catalytically active, which means that the ancient RNA world, if it did exist, it number one, RNA world didn't exist. So let me correct myself. RNA peptide world, if it did exist, it was terminated by the arrival of the protoribosome, which was initially crude in making peptide bonds. But as the ribosome itself evolved around it, along with other minor components of proto life, as we would call it, along with the proto membrane, that led to the emergence of coded protein synthesis, and with that came the DNA, and then came the last universal common ancestor. I hope I didn't muddle up, but that's the current picture. So these 3 papers, if anybody is interested, let me know. I'll be more than happy to share them. These are phenomenal papers. If you can get, I'll try to see if I can get our colleague Dr. Elisa Biondi from Steve Benner's group. Maybe she can come and give a presentation here in this club. I'm telling you, it's an amazing paper. And that group, Steve Benner, is a fascinating scientist himself. He's a chemist by training, and he has actually come up with alternative DNA alphabets, chemically synthesizing them, just which shows to prove, or not prove, it increases the plausibility of DNA alternatives when we look for life on other planets. That's the hypothesis. So I'll be more than happy to share those papers, and I'll see if I can get Melissa on here. Um, she's a very busy person, as we all are. But if she can spend even an hour speaking about the work they do in the foundation, I think it'll be very enlightening for everybody. Thank you.
Well, thank you. I'd certainly be interested in those papers, and yeah, through through some earlier studies and work, um, it is, the whole DNA code is fascinating, and the using mineral surfaces and particularly clays as catalysts. There have been some studies showing that the polymerization is quite feasible, but particularly the importance of the wet-dry cycles in driving the thermodynamics of polymerization through condensation reactions is key. Um, what's interesting, though, certainly in the river shores, is whatever products do emerge, is there's a steady supply of that ultimately to the ocean and through sedimentation to the deep sea. So it is fascinating, and I keep a close eye on the whole question of thermal vents versus river shores and land water barriers, particularly with clays involved. Of how we might piece this story together in a sequence of how that complexity may have emerged. It's all fascinating, and I'd love to hear more talks in this room about origin of life at any of the stages of complexity. It's good stuff. Great.
So,
Yeah, just to give a just to bring this a little bit to my world and to raise for debate something that always fascinates me about this origin of the RNA world and the possibility of the DNA being developed, having evolved through the DNA processes, or if this stuff happened, of if this RNA world was killed and then RNA emerged. One thing that always bugs me with all this theory, probably you guys are familiar with Patrick Fortier, and he's particularly not a big fan of Asgars, but I like his theory about the origin of the RNA and how this migrates to a DNA world. But one thing that always bugs me about this stuff is that there never was found an RNA viruses infecting archaea. We have RNA viruses infecting eukaryotes. We have RNA viruses infecting bacteria, but they were never found an RNA virus infecting an archaea. And this always bugs me because I say, okay, if there was RNA before everything, we have to have these viruses. That's not possible that this is the only ones who got extinct in this whole process. So yeah, just to bring this to debate, that is something that is always on my mind.
well, one speculation on that is perhaps early on they found an effective defense, and the archaeal RNA virus bacteriophages went extinct.
Well,
Yeah, it's possible, but it's really unlikely, I would say, because yeah, this defense systems that you have against viruses in general, they they are really they are really drastic. Usually they kill the cells, right? And so these defense systems usually they do not prolong themselves that much. So that's one of the main reasons why they pack themselves in those defense islands, right? You need more than one defense system, so you can kind of grade it. What I'm trying to say with this is that even if the viruses were extinct, you should still be able to see these defense systems in the cells, right?
it's an interesting point, and it plays into the whole who came first, and there really is evidence that there was complex protein and complex RNA um, evolution going on simultaneously, because there's independent mechanisms for their catalysis and accumulation. And until there's something that is evolved that decides to eat them or is capable of eating them, that, you know, if they would continue to accumulate. And it's a good point that you make, Pedro. You know, why don't we see them?
Yeah, because in extant life, the see, in the prebiotic primordial soup, there may have been n number of multiple pathways leading to so many substrates and products. But what eventually emerged is coded protein synthesis. So if you do a top to bottom approach, the universality and the high levels of conservation point towards the ribosome. It's highly conserved. The ribosomal RNA is highly conserved, and the most conserved part is the PTC. It is almost 99.99% conserved through all the three domains of life. So I just pasted that article that I was referring to as the landmark paper in the chat section. I also back channel to Serena. If anybody else needs it, just let me know. Back channel me. I'll because I do not know who would want it, so I would be more than happy to share that paper. So the RNA world is now being fast replaced by the hypothesis of RNA peptide world. You can also add lipids to them, because without lipids there cannot be membranes. So that is the domain of groups led by Dave Demer and Bruce Demer at UCSC. So it's a prebiotic clutter from which coded protein synthesis emerged. There may have been other alternative systems which are now being tried under laboratory conditions. That doesn't mean that they all survived. So, and it is virtually impossible to know what exactly happened at the time. We can only make an intuitive approach and say these may have existed based on experimental validation of these possible pathways by recreating systems. But eventually, everything led to there was like a bottleneck which reached to the emergence of coded protein synthesis. So that's my take on that. I'll be more than happy to share those papers. Thank you.
This is a fascinating discussion. Thank you all, and especially for Parth sharing the great papers. I'm interested as well. So, I guess the this partially answered part of my earlier question on the which one first. So the for Petro, you mentioned the seems to be a very intriguing fact that there is so far no known RNA virus infecting archaea. But I did found a Google through Google a link to some experimental findings that finding not the virus but the RNA polymerase hallmark. So I'm curious of your comment. So my question for all the experts on stage is what's your take on the role of viruses? So be it uh, parasitic or symbiotic, with the whatever the LUCA, the first cell of life. So they're just playing some sort of functions that they has always been there, coexisting, as old as RNA maybe. I don't know. What's your comment, Pedro and Parth and Pedro, yeah, and others? Any thoughts?
I I I think it's pretty complex. As I was saying before, my personal view that is that everything exists, uh, and at some point, some of these clusters of biological signature things, or I don't know even how to call it. One of these primordial cells, let's call it, or pro cells, I don't know, uh, have that ability to divide themselves, and this is what we start calling cells. And then others are not able to replicate by themselves, but once they integrate one of these ones that could replicate themselves, we're starting to be able to do it, and this would what we call the viruses. But I think nowadays, if we see the function of the viruses, a lot of people say that viruses and bacteria coexist for so long that we cannot call this as a predator and prey relation, because if it was a relationship like this, well, bacteria would be extinct. Put this way, a lot of people have this argument. I don't know how to put this exactly, how to classify what type of relationship. What we can say is that both one need each other, and the type of virus infections, especially that happens in bacteria, so to bacteriophages, it's really important to regulate the amount of bacteria we have in the environment, especially in the ocean, to make the cycling and making the nutrients available again for other cells, and to restore the populations, restore the dynamic of the community. So in a broader perspective on the impact to the ecosystem, the part of the viruses infecting the phages infecting bacteria, the viruses infecting archaea, and the other viruses infecting microeukaryotes is really important for the chain and the equilibrium of different types of ecosystems. And apart from this, on only the killing, let's put this way. So one interesting data that there is in a recent paper, uh, that says that every 48 hours, if I'm not mistaken, I think it's 40% of all bacteria on Earth are killed by phaga infections. So every 48 hours, you have half of the bacteria killed. But of course, through their replication, this number, it's not so important, right? Because in 48 hours you kill half, but in 48 hours you already have way more bacteria presence there. But yes, just want to highlight this. But apart from the scaling part and this type of infections, viruses we know had play a really important role in horizontal gene transfer processes. So to make the genomes of cells evolve in a faster way and became more efficient, viruses play a huge role. We also have that. We all read somewhere sometimes that a lot of our genomes came from viruses, which is not entirely true, but it's the idea is this. The idea is that we receive a lot of genes that were not originally or were not only generated by mutations of our own genome. So these two roles of viruses cannot be forgotten. Like the dynamic they bring to the ecosystem, and also how they speed up this type of evolution through the horizontal gene transfers.
Just a comment, and I do want to check with you again. We've been going 2 hours, and if you're coming up on time, some of these discussions could just keep going. This is a prime candidate. Um, are do we have you much longer, Pedro?
Not much longer, but I don't know, maybe 10 minutes. I can say.
Okay, I'll sneak in one comment here. Um, so it certainly the functionality of the ribosome and proribosomes and translation is a game-changing event for for the original life, and it's curious to, and a fascination of mine to contemplate what happened before that. In part of my interest in clays and the genetic code is noticing, and when I was doing some work on clays, how coincidentally fortunate the geometry of certain hydrating clays like montmorillonite and other phyllosilicates is that a triplet of DNA uh, happens to be geometrically compatible with the stacks in clay layers, and the edges of those clay layers can become phosphatized, and the intermediates of the citric acid cycle that go through diphosphates are compatible with those stacking layers. And a significance of the wet dry cycle, or the wet dry transition, in swelling clays would provide a mechanical force for those to actually activate and catalyze different reactions in those intermediates. But in terms of looking at viruses, there's always the question. Well, so we have proteins, we have glycoproteins, and we have RNA, DNA. The enzymes to produce those components would have to be operative before viruses could really emerge as a operative agent of infection. And the predecessors of each of these functions, as we sort of puzzle together how they may have arisen, may have very different compositions than what was ultimately proved to be more efficient, later and replaced in terms of, the predecessors of cell membranes, the predecessors of, what would bring uh, amino acids in proximity to triplets of RNA or DNA. Clay edges and clay surfaces are at least geometrically compatible and chemically compatible with those requirements for early catalytic activity, and wet-dry cycles could be the ultimate driving force for the changes in favorable equilibrium. But they would be much slower than anything that we would call living. And it's fascinating to sort of try to put that puzzle together in abiotic chemistry as it leads to more of what we would start to call living.
Absolutely. So, Serena, you brought a very important point. They would be so slow. One of the biggest obstacles in the RNA world as well as RNA peptide world hypothesis is the absence of an RNA replicase which can help make copies of RNA. There has not been a single demonstrable RNA replicase that has been found. And that is where what you just said is absolutely relevant and has some very important implications, because the wet dry cycles would have definitely driven something forward, but not at the rate we would want them to. But somehow that is only plausible thing that occurs to me as well as many other people in this field. Stay tuned as more experiments are being done by astrobiology labs around the US and also the rest of the world. Thank you.
Yeah, thank you. I agree. This has been an amazing discussion, and really fascinating. It's not my expertise, but it's fascinating to listen to the discussion. And Pedro, thank you so much for sharing this amazing and so important work to know where we and our nucleus comes from is quite important, and it's still so fascinating to me that we have the tools to look back in time like this. So yeah, thank you so much for this amazing work, and thank you, Parth, Serena, Eli, Frank, Dr. Shah, Dennis, everyone, Nick, Victoria, for the great interview. Thank you everyone for making this wonderful room. And Pedro, thank you so much. And I hope you come back one day and share some updates if you found more clues to all of our questions here. Right. And yeah. Do you get
Yeah, that,
do you get to go on the ships like with Woods Hole? I worked there for a year actually in Woods Hole, but at the Marine Biological Laboratory.
oh, that's awesome. No, I didn't. I haven't been in the lab. So I joined the lab during COVID. So we didn't have any sampling trips since I arrived in the lab. But yeah, I have been in other expeditions, but no, not anyone in Woods Hole. But I would love to. I would love to. And thank you so much for having me. It was really nice discussion. I hope I can come back one day, part with the to describe here to you all RNA viruses infected on archaea, or a more complex part of the story on new insights on how we acquire the nucleus that we have today. But yeah, it was really nice to meet you all and to discuss this our work and other fascinating questions that we have.
Yeah, great. And enjoy your vacation. Enjoy Portugal. And yeah, have natas on me. Thank you. Bye, everyone.
Thanks, everyone.
Thank you,
Thank you.
everyone.
Thank you, Pedro.
Bye. Thank you, Pedro.
Bye.
Thanks,
Thank
Pedro.
you.
Thanks,
Thank you,
everyone. See you all soon.
Karina, and everyone. Thank you, sir, Karina.
3, 2, 1. Bye.